I talk about these things called the four horsemen. The four things that are basically coming for us all. I really should have added a fifth horseman, and that is immune health.
Thank you for bringing this up. There are two organs that are rate limiting in terms of your aging, and it's the central nervous system and the immune system.
What is our strategy for mitigating immune decline?
The proof is in the pudding, in a way that we know exercising and a combination of exercise is the best anti-aging intervention we have.
Turn over to where you wanted to around metabolism.
What is it about metabolism that really is so important? I'm convinced that it is fuel utilization. I think ketones are probably the cleanest fuel to burn in terms of, again, byproducts, oxidative stress.
You're going to put more GLP-1 in the system. You're going to overcome the resistance at the beta cell. You make more insulin. You now have better glucose control. Everybody wins.
I just wanted to really experiment for myself, just try to see, okay, what is this drug really doing? And it's been nothing short of remarkable, I think.
How could it be that supplementing NAD does not lead to a longer, better life or some health benefit? Hey everyone, welcome to the Drive Podcast. I'm your host, Peter Attia. Eric, thank you so much for coming to Austin. I know it wasn't just to talk to me. I know that half of it was getting you to drive on the track at COTA tomorrow with me. So we're going to have some fun there.
My pleasure.
But as much as I think the two of us could sit here and talk about race cars for the next 3 hours, I don't think the audience would appreciate it or care for it as much as they will care for what we will talk about, which is your work in geroscience. So maybe give folks a little bit of a sense of what attracted you to this field and how your journey and background brought you where you are.
It's a bit of a serendipitous type of story in a way that I'm an MD by training from Belgium, did my last year of medical school at Harvard. And this just sort of opened my eyes to a whole world. I was the first person in my family to go to college, so this was, ending up at Harvard with some of the best teachers, some of the best students was just mind-blowing. And I went to medical school wanting to do research. I never had that sort of a doctor fiber, I call it, so really wanted to actually do research. And so after this, finished medical school and came back for directly a postdoc at the Joslin Clinic working on diabetes and metabolism. And so this is where the story gets circuitous. Ended up becoming interested in the reason for the etiology of type 1 diabetes and worked on viruses and autoimmunity. This eventually led me to, mostly a career in virology, which confuses people. So I spent many years working on a variety of viruses, including HIV and herpes viruses and so on. And through that work, we ended up cloning a family of protein called some of the first epigenetic regulators, the HDACs. And the HDACs at the time, that was 1996, we were responsible for the cloning of a whole family of these epigenetic regulators, ended up being important in aging. And starting in around 1995, 1996, my lab slowly shifted towards the study of aging. And to this point today, actually, I only have one last postdoc in the lab who's working on HIV. The whole lab is actually focused on epigenetics, immunology and metabolism, so that the interface between these variables. So in some ways, it's the beauty of an academic career, which I've just followed my interests and sometimes followed the money a little bit in terms of funding. Yeah. And now, I have another additional responsibility, which is to lead the Buck Institute for Research on Aging. So I split my time between the lab and some more leadership type of activities.
You mentioned two things there, metabolism and immunology. Talk a little bit more about how each of those individually contributes to aging. I think most people will intuitively understand it, but talk maybe a little deeper about it.
Well, first, immunology is central to aging in many respects. One of I hope we can get to talk about this later. There is data showing that there are two organs that are rate limiting in terms of your aging, and it's the central nervous system and the immune system. And the reason for this is actually one could have predicted this based on the fact that both organs are distributed organs. If you think of your immune system, it's located in pretty much throughout the whole organism. And so its activity can influence the well-being or the functioning of every single organ. The same goes for the central nervous system. And there's a recent study coming out actually from the lab of Tony Wiscoré showing that those biomarkers that measure aging in those organs appears to be the most predictive of your lifespan. There's also incredible data showing that if you induce a specific lesion in the immune system, for example, in mice model, if you knock out ERCC1, DNA damage repair, only in the bone marrow, so that the whole immune system is affected, you actually induce accelerated aging in the whole organism and senescence in every single organ. So there's the immune system.
In what model?
It's been done in two different models in mice. So it's been done with the ERCC1 mutation. It's also been done by knocking down the major TFAM, the major transcription factor for mitochondria. So if you induce mitochondrial dysfunction only in the immune system, you induce secondary senescence in the whole organism.
Do you think that would be true in mammals? In humans?
That's always that's $1 million question. In some way, it's been shown in two different models in mice.
B6, which?
B6. I don't remember the exact strain of the mouse, but there's no reason why it should be different, frankly. And it speaks to the importance of the immune system. The second way for the immune system is through chronic inflammation, which is, tied cause and effect in the whole aging process. Now, and we can talk about this later as well. I find it fascinating, the whole idea of chronic inflammation, which is induced by the aging process, but which itself actually further accelerates aging. So there's really a lot of work that's being conducted in this area. The other one that you were asking is metabolism, I think is...
Can I ask you a question before we go?
Yeah.
That's a very interesting idea that two organ systems that are going to be rate limiting in age are the central nervous system and the immune system, both of which are distributed. Where would you put the endothelium in that list as well, right? The endothelium is also quite distributed across the organism. And do you think that there's an inevitability to basically endothelial damage as a process of aging, which of course results in the leading cause of death, the atherosclerotic diseases? Do you think of it the same way, or do you think of it as different?
It's not sort of defined as an organ by itself. It's a cell type, but I agree with you, it has incredible importance, and especially as it affects the heart and the cardiovascular system
and the brain.
Brain. But I think of it as not so much as an organ, but more as a sort of a principle that maintenance of barrier function in not only in the endothelium, but also in the skin, in the blood-brain barrier, are emerging as key areas to focus on if you want to maximize your longevity.
Yeah, I want to come back to this in great detail, Eric, but let's, for the sake of summary and synthesis, turn over to where you wanted to around metabolism.
So metabolism is essential to life expectancy. If you look at, and for a number of reasons, and one of them, I'm convinced, even though that theory has been somewhat discredited, the whole oxidative stress theory of aging, I still think oxygen is one of the major problems associated with the aging process. We have not been able to target the oxidative stress using antioxidant. That has failed. It doesn't mean that the whole oxidative stress theory of aging is not valuable. I think living in an oxidative environment is really, is one of the mechanisms that leads to aging. Not the only one. Aging is pleomorphic.
But just to make sure folks understand what you're saying, Eric, you're saying that that the generation of free radicals through oxygen, so I don't know how technical we want to get for people, but I think unfortunately we might need to get a little more technical, and apologies to those who don't want to go this deep, but we have to talk about kind of what the role of the electrons are in oxygen and why free radicals form and what they do. So maybe we do go a little deeper here and explain what you're saying, because it's a very important concept, and I think we should probe it.
Well, I don't know how much. I mean, maybe you'll do a better job at explaining this in for the lay person. I mean, oxidative stress is the fact that pretty much the main metabolic reaction are dependent on oxygen, which gives its electron. It's in the so-called respiratory chain. There is leakage of these electrons that are traveling down this respiratory chain, leakage at specific places. If the process was 100% efficient, the whole energy would be transferred from metabolites such as fatty acid, glucose, and so on. But it turns out the mechanism is actually leaky. These electrons reacting with oxygen can generate these byproducts called radical oxygen species, which are highly reactive.
Right. So they're not chemically stable the way we think of a normal atom of oxygen.
No. And so they tend to react with proteins, with fatty acid, and they induce lesions. And we have a whole the importance of this system in terms of protection against it is highlighted by the number of molecular systems that we have that are actually protecting against this.
And we know that as we age, that leakage increases.
Exactly.
So something about the integrity of the mitochondria and the respiratory electron transport chain degrades as we age. And therefore, we see more and more of this leakage, yeah?
Yes, absolutely. And so out of this came the whole idea, well, let's just suppress oxidative stress. And, there are chemicals, even some as simple as vitamin E, vitamin C, that you could imagine that by chemical knowledge would be predictive to be able to quench these radical oxygen species. And so a number of...
So the idea, sorry to just keep interrupting you, but we'll play off to each other to do this.
This is
so
great.
You eat, for example, an antioxidant, and as you said, it neutralizes that reactive oxygen species with its unstable electrons, kind of like you would throw a blanket on a fire that's simmering.
Exactly, and that was the hope. So when the theory was proposed, a whole industry actually grew up out of this, the whole antioxidant and the antioxidant diet and the, the vitamins and so on. You can still, by the way, that whole industry is still existing today.
Sure does.
Now, what happened is that when clinical trials were conducted in this area, they failed. And so, people who think relatively simply decided, well, the antioxidant failed, therefore the theory has no validity. And I would say not so fast, because it turns out that these radical oxygen species also have important roles. They actually are inducing an inflammatory response, which can be protective. And a good example is during exercise. There is some evidence of activation of oxidative stress during exercise. And if you neuter this, for example, with anti-inflammatory, you probably remember the data showing that anti-inflammatory drugs tend to sort of suppress some of the beneficial effect of exercise. Yeah. It's the same whole idea. And so this is one case in which these radical oxygen species can have a protective role and actually a signaling role. So when you suppress it completely with these sort of global non-specific antioxidant, essentially you're not only killing the bad guys, but you're also suppressing an important signaling mechanism.
There's another hypothesis that I would offer, which is it possible that there's still a net negative to the free radicals? So there might be some benefits, but more negatives. But it could be that the trials were using agents that were simply ineffective, because the problem is we don't have a great biomarker for the state of free radicals. So it's sort of like saying, I have a hypothesis that this biological process is bad. I can't measure it really, but I think it's bad. I have a drug that I think will tamp it down. Let's give the drug. The trial failed. Well, do you actually know if it tamped the thing down? We don't even know if we tested the hypothesis, correct? And so those would be kind of two distinct plausibilities.
I completely agree. And it's quite often the case is that, the whole story of vitamin D is a good example.
Absolutely.
Where people will tell you, vitamin D doesn't work because they conducted clinical trials, but they didn't, they didn't adjust the dose, they didn't measure the levels. So it's a bit the same story. There are markers that you can actually use in research environment, like 5-hydroxynonenal or protein carbonylation, which are indirect markers of lipid or protein oxidation.
And how efficacious or beneficial, or I guess the word is maybe, how complete are they in the scope of understanding? And have we demonstrated that mega doses of vitamin E or vitamin C will indeed suppress those markers in humans?
They're not great.
Okay.
They're not great. I had a colleague at the Buck Institute, Martin Brand, who is one of the leaders of the whole mitochondrial field called bioenergetics, which is the study of how the respiratory chain and energy metabolism happens in mitochondria. And he came up with the idea that he identified many of the sites where these unique radical oxygen species are generated. And he was able to generate specific inhibitors for each of the sites and was able to show that actually inhibition at some sites was beneficial, while inhibition at other sites was not beneficial. And so this project was actually supported by a pharma company, which eventually decided to drop the program. And he's retired, which I think is a great loss, because I think this is a whole program that still needs to be pursued.
So if I'm understanding what you said correctly, Eric, it sounds like there's a much more nuanced view. It's not that free radicals are bad, and it's not that free radicals are good. It's like everything in biology. It's the Goldilocks rule. You might need more of it during this circumstance in this part of the body. You might need less of it in this circumstance at this totally different part of the body. And as a result, any strategy that would try to globally suppress it could, even if successful in doing it, which we haven't been able to measure, might actually not yield to a favorable outcome.
Totally correct. Totally correct. And I get frustrated by these types of the way that people sort of love to oversimplify or, sort of erase whole fields. We've seen this, and I suspect we will get to talk about sirtuins, because the same thing has happened in the sirtuins. There's a lot of amazing work done, and then a few negative results or things not working out. NAD metabolism, same thing. And I always tell people, once you get into any field of study and you go deep and you start testing in humans, put on your seatbelt, because it's not easy, and there are no magic bullets. But I think stopping the study and saying the whole field is BS is really, for me, not the way to go. We got to dig deeper, and eventually we'll get to that.
So tell me what else within metabolism you think is kind of a hallmark of aging. So we obviously talked about the central part of metabolism, which is respiration and ATP generation and the leakage that occurs there. And basically, unfortunately, that just appears to be inevitable.
Yes, that's totally right. We will never stop the oxygen in our environment. So
yeah. I do like to tell my patients that this is why I kind of harp on them to do a lot of zone 2 cardio training, right? So zone 2 very specifically, by definition, right, is the canonical exercise you would do to maximize fat oxidation, which of course implies the most efficient use of the mitochondria. And the hypothesis, because I don't think, we don't have proof of this, but the hypothesis is training at that level for specific periods of time throughout the week is a way to improve the health and function of your mitochondria, which would hopefully imply that you're reducing that degradation of function. Do you think there's validity to that, at least, first order logic?
Yeah, I mean, the proof is in the pudding, in the way that we know exercising and a combination of exercise is the best anti-aging intervention we have.
But do you think part of it is through that exact mechanism?
Yes.
Yeah. I mean, that's
likely.
been my hypothesis. But again, we can't fully glean that in any human clinical trial.
No, hard to study. And I think, your point allows me to address your question. What is it about metabolism that really is so important? I think I'm convinced that it is fuel utilization. And you mentioned, fatty oxidation versus glycolysis, and I'll add ketosis to this. I think if you think about your metabolism is able to oxidize a number of different substrates, amino acids, fatty acids, glucose, and ketones.
And lactate.
And lactate.
Yep.
And every one of those actually burns with different efficiency. Both being car aficionado, I think your audience probably knows also that, there's different to burn diesel or to burn 100 octane gas. And, if you look at that hierarchy, I think ketones are probably the cleanest fuel to burn in terms of, again, byproducts, oxidative stress. They seem to be really unique in this
how would
respect.
you rank order from cleanest to dirtiest, inclusive of lactate?
Lactate, I would not be able to put it.
Okay.
I think it's probably clean.
Yeah, I
would
think
bet
my
it-
intuition is it is as well.
But it would be the top would be
Beta-hydroxybutyrate.
Beta-hydroxybutyrate. Acetoacetate is present at such low abundance, it's probably not relevant as a fuel source. Then fatty acid. Next is the worst is actually glucose. And when you think about metabolism and aging, for me, it goes to a lot of the data that has emerged from the ITP, for example, intervention testing program. This
Rich Miller has been on several times.
Yes, I watched your recent podcast with Rich and others. One of the remarkable thing when you look at the drugs that have 7 or whatever 10 drugs that have emerged out of 80,
Yep.
they are really targeting glucose metabolism, and very via completely different mechanism. Think about acarbose, which is blocking, absorption of glucose. Think about the canagliflozin, which is, targeting a protein that has nothing to do with links to glucose reabsorption in the kidney. Think about metformin, which is,
But metformin failed.
Yeah, it failed, but it seems to be having very powerful effect in terms of, it did not fail actually.
It failed unless it was paired with rapamycin.
Yes. And in monkeys, there's a study coming out that showed that it actually had an effect on lifespan. So,
And do you think rapamycin has any impact on glucose metabolism favorably?
generally, actually, rapamycin is the exception to this because it seems to be having some. It's not indifferent. It has been claimed to be having an effect on insulin sensitivity. Although
I'm not clear if that's true at the doses, but anyway, we can come back to that.
I've taken rapamycin. I have not seen any effect on my blood sugar. Now, so think about, acarbose, canagliflozin, metformin, and now the GLP-1 agonist, which I predict will emerge as geroprotectors in the future. So I think that's really speaks to an important aspect, which is fuel utilization. And how, whether you're burning a clean fuel, whether you're burning a dirty fuel. We've put, for example, mice on a pure fat diet. These mice never saw a carbohydrate during their life, and they lived longer, which I thought was actually quite interesting.
It is interesting, Eric, because a lot of the mouse literature, I think people don't read the fine print very closely, and they don't notice that the typical thing you'll see is these mice were fed a high-fat diet to induce obesity so that we could test, drug A, B, or C against obesity. But in those studies, it's not just a high-fat diet. It's a high-fat, high-sugar diet, right? So they're making some insanely hyper-palatable, the closest I can come up with is they're making a donut.
Yes.
Right. It's a fried dough sugar food. And, so they're making basically donuts for these monkeys. And that's different than saying it's a high fat thing. So yeah, I think it's important to point out because, a high fat minus the sugar might not be the same issue, right?
I agree.
At least in that model. Yeah. So what do you think it is about glucose metabolism that leads to this? Because for all intents and purposes, let's just go through the metabolic pathways, right? So glucose, 6 carbons, it gets broken down into pyruvate. You get your 2 pyruvates for 1 glucose, right? And then pyruvate let's just assume we're doing this under aerobic conditions, so we're not in a rush. We're going to take those pyruvates. Do they turn into acetyl-CoAs? I can't even remember.
Yeah.
To then enter the...
It's actually one pyruvate, and that leads to, enters the mitochondria and becomes acetyl-CoA.
Acetyl, okay.
Yeah.
So what is it about that process that is not as efficient as when you are cleaving off carbons from a free fatty acid and those carbons are turning directly into, I think, just a straight acetyl-CoA and then entering the Krebs cycle? What is the, I mean, it's a very subtle difference. Why is one so much more inefficient?
You mean, why is there more calories per fatty acid?
No. That can be
yes.
explained by the stoichiometry. Why is one, quote unquote, dirtier?
Okay.
Yes.
I think, obviously, this is a really complicated question.
Yep.
So I don't know that I would be able to really tell you purely as fuels whether there is a difference. I think the biggest difference is in terms of the whole mechanism that they elicit. And when we think about glucose, I don't think necessarily of it, if you were to study it in a tissue culture dish, that one would be more toxic than the other. I don't think there's any evidence for this. But glucose, and particularly the form of glucose that we have not evolved to actually be exposed to, which is all the wheat products, this fast form of glucose, elicits insulin secretion. And I think insulin and IGF-1, but particularly insulin, is the culprit in this whole process.
So you're not saying that 1 mole of glucose, 1 mole of free fatty acid, we know there's a difference in ATP generation, but you're not saying that there's a difference, assuming they're both going through the mitochondria, you're not saying there's a difference in free radical formation mole per mole? Or are you saying that it's this way? There's another way to explain it, which is per mole of ATP, you need to run so much more glucose through that, of course, you're going to get more leakage.
Well, the key difference is that the glucose is generating ATP not only via acetyl-CoA and pyruvate, but is also generating ATP in the intracytoplasmic components.
Right.
The fatty acids do not generate any. Right. So I suspect that there might be a difference in terms of the amount of free radicals that are generated. There is evidence, but I would not be able to cite you the paper, that one burns more cleanly than the other. And I suspect it's partly the cytoplasmic component of glucose. It's also less efficient in terms of the amount of energy that's being generated per gram of fatty acid, or per mole of fatty acid versus per mole of glucose.
And then going back to the insulin IGF component here, what role do you think they're playing?
Critical. Because epidemiologically and through studies, we know that the insulin response to your glucose. So if you do a lot of sports, I'm not a proponent of the low carbohydrate or no carbohydrate diet, because there's very little evidence that those diets are actually beneficial. I gave you the example of a ketogenic diet, which we did experimentally, but these are not practical diets for anyone.
Just because of the challenge in avoiding carbohydrates in the standard world we live in?
Yes. I would say both socially, palatably. I mean, there's so many reasons. I went on a ketogenic diet. From what I remember, I think you went too.
I did for 3 years. I was on a ketogenic diet, so we should compare notes.
Yeah.
Because I want to hear your experience, and then I want to ask you a couple of questions about it.
It was very hard.
How long did you do it?
For a couple of years, and I did not feel super healthy, which is really kind of interesting. I found it sort of socially isolating. And so we've worked actually to remedy this on. We can talk about this later on a novel keto.
Like a ketone ester?
A ketone ester, exactly, of beta-hydroxybutyrate. So going back to the role of insulin, there is a lot happening in terms of, that's been documented, that the intensity. So first, your average glucose plays a role, average blood glucose. This is measured by hemoglobin A1C in a whole series of complications, cardiovascular, as you know. But perhaps more important is the intensity of your peaks. And I think, the intensity of the peaks of insulin is a reflection of your glucose intake, fast absorbing glucose. And that has that's the reason why I advocate people to, go on a CGM, continuous glucose monitor, and to really learn to understand what spikes them. The whole idea is to mitigate these peaks of insulin secretion.
I'm just going to play this for all of our patients because we have this discussion with every one of our patients. So it'll be nice to just play this video and let you do the talking.
The whole idea there is to mitigate these peaks, and either dietarily or, for example, the GLP-1 agonists are playing a role in this as well.
Let's talk about this because there is, at least for me, a great deal of confusion around this point. Now, we understand today the role that the gut plays in metabolism, and we understand that a lot of it is transduced through GLP-1. So endogenous production of GLP-1, according to Ralph DeFronzo, the world's authority on this, is what's driving 80% of beta cell activity with respect to insulin. And therefore, when we have insulin resistance, the GLP-1 we're making is insufficient to generate the insulin that's required to manage the glucose. Makes sense if that's the case, that giving exogenous GLP-1, you take a shot of tirzepatide or semaglutide, you're going to put more GLP-1 in the system, you're going to overcome the resistance at the beta cell, you make more insulin, you now have better glucose control. Everybody wins. Now, it's not clear that that has anything to do with the weight side of it, and that's a separate issue, and I want to actually talk about that because there are two very interesting theories as to why these things cause weight loss. But the point here is, wouldn't you expect to see higher levels of insulin in someone taking a GLP-1 agonist to achieve that better glycemic control?
Yes, and that's not what you see. I don't have an answer for this, but I've seen the same thing. Including personally, I've been experimenting with tirzepatide. My insulin is 5 now, which is lowest that you can possibly get it. And there was a part of me that was worried that I was going to go against my own whole theory about
Have you checked postprandially? Have you done an oral glucose tolerance test where you look at? Because that might be something to do to see what is happening to postprandial insulin along with postprandial glucose, which of course will be better.
No, I haven't.
That would be an interesting test to do, yeah.
And I've worn a CGM. My A1C has gone from 5.4, 5.5 to 5.0.
Yep.
And my insulin is down to 5.0 as well. So.
And did you lose any weight?
I lost a little bit of weight, not a huge amount. 6 or 7 pounds, which was never the goal to start with. It was the whole idea was and no loss of muscle mass, which actually is the big boogaboo that people will have you fear. No loss of muscle mass if you are exercising. So for me, it's an experiment. I haven't decided this is something I'm going to continue, but I just wanted to really experiment for myself to try to see, okay, what is this drug really doing? And it's been nothing short of remarkable, I think, in some way. One of the most surprising has been for me this feeling of satiety. You hear about satiety. I was never in my whole life the type of person that felt full. I could always eat more. And all of a sudden, after about 2 weeks on this, I just looked at my plate and said, I'm full. And I heard myself saying this, and it just felt like, well, this is really completely different. And for me, the reason why I'm excited about these drugs is, and by the way, this is not an endorsement. This is something to decide.
Yeah. This is self-experimentation,
self-experimentation, which is a long part of the tradition of our field, which I love. And the whole idea is really to, for me, the thinking was one of the biggest advance in longevity medicine is this idea that a range is meaningless. And as a practicing physician, this. I went to medical school and we were told that, your blood pressure has to be 130 over 90. And that was still a normal range. So you could be 128 over 88, and you were still considered normal. The same thing, I went to see my personal physician and told him, my blood sugar is creeping up every year that I'm doing it, and now it's 96 fasting blood sugar, and I'm worried because soon I'm going to be
And he said, it's below 100, it's okay.
He told me, you're normal, don't worry. And I told him, I said, what is normal? And I think this really is where I think longevity medicine is going to make an important impact, is really sort of revisit.
I can't tell you how many times I've had this argument with people about glucose. And here's the funny thing. We have the literature. In other words, we have literature in non-diabetics.
Your A1C.
That says the lower the A1C, the lower the all-cause mortality. It's a monotonic reduction that knows no lower limit.
I'm with you.
So we say that up to 5.6 is normal. And if you're at 5.6, you're fine. And 5.5, but 5.5 is better than 5.6, and 5.4 is better than 5.5, and 5 is better than 5.4, and 4.8 is better than 5.1.
I'm not there yet.
Yeah. But my point is, I also find it, I don't know what the word is, maybe sad. I find it sad that we've simplified this problem in an effort to communicate, but have lost the essence of where is lower better. Because it's not always true in biology, right? It's not, when you look at TSH, for example, when you look at thyroid hormone, much more narrow band in which we would say there's optimal. And lower is not, if it's too low or too high, it's problematic. But it turns out that when it comes to average blood glucose in a, non-type 1 diabetic or someone who's taking insulin, under natural physiologic circumstances, it's just better to be lower. And as you age, it just keeps creeping up.
Same thing for blood pressure.
Yes.
They're revisiting the number every 5 years in terms of making it lower. I think if your blood pressure is 105 over 65, you're better off than if you're 115 over 75.
That's right. Provided you're not symptomatic, lower is always better.
And so I think it's, I'm frustrated, but I'm also excited by the fact that this is now becoming the norm in a whole new field of physicians who are more aware of what is health. And the same for your weight. We know that it's that's the thing that is really interesting in the whole aging field, is this idea that everything is a J curve. So there's a sweet spot where you want to be, and quite often it's broad enough that you can maneuver this in a way to optimize people's health.
What do you think is the relationship between, body weight is so crude, but maybe we can even talk about it through adiposity, body fat, and longevity, once correcting for metabolic health. So it's obvious that so much of the relationship we see between body fat and poor health is really just a proxy for something that's harder to measure, which is metabolic health. It's very easy to measure body fat, and we estimate body fat from BMI. And so that's why we have all these population data from BMI. But if you have the luxury of working with actual patients, I don't know, I couldn't tell you the BMI of one person I take care of, but I know everybody's body fat, everybody's visceral fat, and everybody's oral glucose tolerance test. So we know what we know, and we know what matters. Are you convinced that adiposity per se is problematic? Or do you believe that a person can have excess body fat but be metabolically healthy and confer the same longevity benefit as a metabolically healthy lean person?
We know there are people who are considered overweight who are metabolically healthy.
Yes. Easily 20% in my experience.
Yes. And I'm not where I think these are facts. No one can dispute them. You can be overweight and metabolically healthy. What I worry about is the long-term effect.
Do you mean from an orthopedic perspective with the other complications that come from excess weight? Or are you saying that they're basically increasing their probability of eventually going off the metabolic slide?
Both. I honestly, I don't know what the data says, but my worry would be that you might be metabolically looking healthy when you're 40, but if you sustain this for 20 years, clearly
Yep.
Visceral fat.
Yep, different category.
Is highly predictive of everything. The other thing I'll say also, the BMI itself my BMI is at the border of being overweight.
I am overweight by BMI, I think. I'm by 4 pounds. If I lost 4 pounds, I would get down to a BMI of 25.
Yeah, and I have 11% body fat. So I don't worry about it because I know, all in all, I'm metabolically healthy. My numbers are good and all this. So in some way...
Yes, it's not a particularly helpful... No, it serves its purpose at the population level, but it can't be used to make a decision about an individual at all.
Exactly. But it can also sometimes... become a confounding variables. And when people do studies and they use these numbers and they make predictions or they draw conclusions that are really not based on the fact that high BMI fraction of the population is heterogeneous in terms of metabolic health. I think this is really where... so my colleagues at the Buck, Nathan Price and Lee Hood, have actually published a paper.
Wait, I didn't realize they were at the Buck.
Yeah, both of them.
They were up in Seattle before, weren't they?
Yes, we recruited both of them actually in the last 2 years. So
congratulations.
Thank you. I think this is transformative for us.
Fantastic,
and really
guys.
Exciting. Lee is still partially in Seattle, so he's partially at the Buck, but he's brought, we've established a collaboration with Phenome Health, and Nathan was at Thorne, and still a CSO at Thorne, but a faculty member at the Buck. So I think and they're really helping us to do something really exciting along these lines. For example, they had a paper describing this BMI, but biochemical BMI based on biological markers that essentially assess, your metabolic status. So I think that, those tools are available, and it's a question of educating the physicians.
And do you know what makes up that biological BMI?
No, but I'll give you the paper.
So, okay, we spent a little more time on metabolism than we did immune health and the immune system overall. I'd actually like to go back and talk about it a little bit more. I think, again, the listeners of this podcast are very familiar with the metabolic stuff. We haven't had as many discussions on the immune system. Talked about it at length with respect to cancer. Had Steve Rosenberg on a few years ago. That was a fantastic discussion explaining the role of the immune system in cancer, which I think we're going to have to talk about here because, I certainly feel convinced that a big part of why cancer incidence goes up exponentially with age is the declining immune system, not just the accumulation of mutations, although I imagine they both play a role. But I will tell you something else, Eric, which is, I wrote a book a couple of years ago about this space. And in the book, I talk about these things called the Four Horsemen. And I describe them as the four things that are basically coming for us all. If you manage to outlive youth, right? So if you're, this is not to diminish the role of trauma and other things that are deadly. But for many people living in OECD nations, it's going to come down to ASCVD, cancer, dementing and neurodegenerative diseases, and metabolic diseases. And people often say, Peter, is there anything you wish you'd written in the book that you would, if you go back in time, you would do? And I say, yeah, there are probably many things if I thought about it, but the first thing that jumps out is I really should have added a fifth horseman, and that is immune health and the types of infections that ravage people in old age that a young person wouldn't even laugh at would laugh at, right? So
thank you for bringing this up because the immunology field somewhat, immunology and aging have been not really mixing very well. One problem is that immunology is an extremely complex and advanced field, probably along with neuroscience, one of the most complex. So when you go to an aging meeting, there is no one talking about immunology. You go to immunology meeting, there are very few people talking about aging. And we try to navigate, even the nomenclature is being used differently. People in immunology talk about immunosenescence, meaning aging of the immune system. They don't mean senescence the way we talk about it in the aging field. So that yields all kinds of crazy communication problems.
Yeah, because if you're in the aging field and you hear immunosenescence, you think of SASPs and things that are being secreted by T cells.
It just means aging of the immune system. Now, the reason why I think this is a tragic failing for both fields is what happened during COVID. became obvious that being infected was not your risk of infection was not linked to your age. The virus infected everyone across, but the outcome could be completely different with 84-fold excess mortality if you were above 75, 84-fold. Now, when this happened, and we can go in terms of trying to understand why did this happen, what are the reasons for this. I went and started to look at the literature. Influenza, it's exactly the same thing. RSV, same thing. So all of these viruses that are, that you can contract in later years will kill you, with really significant rates. Influenza, I think 30,000 people die every year from influenza. The mortality in terms of COVID was really highly segregated into the older part of the population, or in that part of the population that showed accelerated aging, obesity, and so on. So I think there's a lot there.
So do you think that most of the mortality, any time we saw a gap in mortality, whether it was young versus old, whether it was obese versus non-obese, diabetic versus non-diabetic, because any time you looked at that, you saw a difference in mortality. Do you believe that it was always a difference in immune function? I mean, with young versus old, it's very obvious, but do you think that was also true in the other comorbidities?
Yes. Yeah, I would say so.
Yeah.
And it comes from two reasons. One is, there are two broad immune systems, what we call the innate and the adaptive immune system. I don't know if you want me to.
I would. I actually was going to say, I think it is worth going full bore on this. I think it is time for people to roll up their sleeves and understand arguably the most interesting system in the human body. I am biased. I spent 2 years at the NCI doing immunology, but I think this is such an interesting field.
Yeah, so our immune system is built to recognize foreign elements. That really is why it evolved. It has two lines of defense against microbes, bacteria, viruses, fungi, all of those. We are constantly bombarded by those. It is actually amazing because, the evidence of this is if your immune system doesn't function, the bubble
It's incompatible with
life. So we are, we're colonized with bacteria in and out, on our skins, everywhere. So we constantly respond to them in an appropriate manner.
And we survive everything, including disruptions to the barriers.
Absolutely. And so we have two lines of defense in the immune system. First, the so-called innate immune system, which is your macrophages, your dendritic cells, but also pretty much every cell has a whole series of mechanisms that are not pathogen specific. That is, they will recognize an intruder, be it a virus, be it a fungi, be it bacteria, and it will activate a first line of defense. Those line of defenses are non-specific, and therefore they're less effective. And they give time to the so-called adaptive immune system, which is the second part, which is made up of T cells and B cells. And both of those cells have a highly selective defense mechanism. The B cells make antibodies, which will go recognize a bacteria or a fungus or a virus. And the T cells, which are able to actually kill the infected cell itself. So it will recognize when the cell is colonized by a foreign pathogen and will kill it. So the time course of these is that once you encounter a pathogen, you will activate your innate immune response. Typically, it can be fever, it can be all kinds of symptoms, but activation of this defense, and this gives the whole organism a couple of weeks to actually build the defense for the specific that will specifically recognize this organism.
Let's talk a little bit about memory within that system.
The beauty of the adaptive, so the innate immune system does not really have a true memory. It will always react in the same way, no matter how many times you encounter the.
So the same, if you get the same virus. So if your kids are ping-ponging the same respiratory virus at you from school,
yes,
your innate immune system has the same playbook.
Yes, fever.
You're going to get red. You're going to whatever,
inflammation.
Yeah, you're going to get sore.
Yes.
All of those things are going to happen regardless.
Exactly. Yeah. And that's in contrast to the adaptive immune system, because once the initial response has been generated, either via an infection or a vaccination, this is what a vaccination is. It presents you with a given fraction or the whole virus or a part of it. Your body will mount a response, and this will lead to the amplification of a subset of cells that are selective. So think about either your T cells or your B cells. None of them are the same. We have a process by which we generate so-called diversity, which is billions of different forms of antibodies or T-cell receptors that are recognizing, in principle, every chemical structure or every protein from a microorganism. Now, what happens during the initial encounter, either be it a vaccination or an infection, is those B cells or those T cells that have a receptor that is able to recognize the pathogen will become amplified. And they will churn out large amount of the antibody or the T-cell clones. Once the job has been done, they will contract, but they will not contract back down to the same level. They will become what we call memory T cells or memory B cells. So that if you encounter the same antigen in the future, the reactivation process is shortened, the maturation happens faster. So eventually, that's the whole idea of the vaccination, is to sort of get yourself ready with a subset of memory T-cell clones or B-cell clones that once the true virus will come, you will be able to mount a response within a few days or up to a week. And so that's how vaccination works. Now, what's interesting during aging is, and people are not aware of this, if you're above 70, most vaccinations do not work. And so people then will ask, your immune system has aged and your vaccination rate really decreases very strongly. From what I remember, this might be different in different populations, but the vaccination rate success is close to 30% if you're above 70.
Now, during COVID, what was the risk reduction for a person over 75 who was vaccinated versus not vaccinated?
Oh, it was almost complete reversal of the effect in terms of the protection.
Meaning it was highly protective.
It was protective.
So how do we reconcile those two facts?
That's true. And I don't know, to be honest, I don't know how this has been studied. I don't know. I would be happy to read about this.
Okay. Because the COVID vaccine seems to have had a remarkable risk reduction in very old people. Didn't seem to have an impressive risk reduction in younger people because the absolute risk was so low. It didn't seem to matter that much. But boy, did it matter in older people.
But did it matter at the population level or at the individual level? This is what I'm not sure about.
I certainly don't want to go on record saying something that I
it's the same for me.
But my recollection, I mean, I think we can find the answer and put it in the show notes. My recollection, which could be wrong, is that the older a person got, the greater the benefit they got from COVID vaccines with respect to mortality. So I guess the question is, let's maybe talk about other vaccines. Is that not the case with influenza? Is that not the case with pneumococcus or any of the other vaccines that are used primarily in older adults?
In general, at least, and I'm not a vaccine specialist, but the thinking is that there's a dramatic decrease in the efficiency of vaccination against influenza, against RSV, against all of those as you age. The thinking then is how does it work at the population level? And this is where the whole concept of herd immunity works, is that if you limit the spread of the infection in a family, for example, you're much less likely to infect grandpa who will.
I see.
So that's been my understanding of how most of these viruses, vaccines work.
I'm asking a different question. That's an important question. I guess I was asking obviously they didn't probably do a randomized control trial, so you've got all these confounders in it, but I wonder if they just looked at all comers to the hospital vaccinated versus non, let's try to control for all the confounders. If the hazard ratio is 1.2, it means nothing, or 0.8. But if the hazard ratio was, 0.2 or 8, well, you'd say even with the confounders, there must be some high degree of protection that came from that. So anyway, I'm sure someone listening to this knows the answer to that. We'll try to find the answer and put it in the show notes. But so let's go back to the why, right? Why is it that as a person ages, they're less likely to respond to a vaccination? Is it because A, their immune system, the adaptive immune system, is less able to recognize the foreign pathogen and build up a high enough reserve of T cells and B cells that will respond? Or is it B, that they can do that, but the ability for those cells to stay in a memory state and be reactivated is somehow impaired?
I think it's both. As in everything in aging. Aging is, but there's one aspect which is really unique, at least in terms of T cell, which are really instrumental in terms of most vaccine response, is the fact that these T cells are generated, the diversity of the T cells is generated by the thymus. And the thymus, a small organ behind the sternum.
How big is your thymus and my thymus right now?
I'm 68, so it's probably very, very embryonic, and there's probably not much left. So after age 50, in most people, you find it very small. Now,
whereas when you're young, it's actually, you can see it on an imaging study. Yes. And I would imagine if you and I had a CT scan of the chest, you'd barely be able to pick it up.
Exactly. And it's replaced by fat, actually, in most people as you age. Although there is some somewhat controversial evidence that there might still be some clones that can be reactivated even in older people. And human growth hormone, as you know, is one of the intervention that has been shown to actually reinduce thymogenesis.
So let's talk about that a little bit. Are you referring to that Fahey paper from about 7 or 8 years ago that looked at growth hormone with metformin and DHEA or something like that?
That's one, but that Fahey paper was actually inspired by work of a colleague of mine when I was at the Gladstone Institute, who did this, actually Mike McHugh and colleagues did this in patients with HIV, who were chronically infected with HIV, where they lose a lot of their CD4 T cells. And there was an interest in trying to see if you could actually, and so there's a big lesion initially in infection, and there was an attempt to actually try to see if you could regenerate these populations to bring them back to our normal. Because even though we had great drugs against HIV, they could not bring those patients back to normal. There was a remaining sort of original insult. So they did a trial with human growth hormone that were able to show some degree of thymogenesis and increase in naive T cells in these patients. And I believe the Fahey trial actually tried to reproduce this. I think there's a second Fahey trial that is ongoing, but I haven't seen the results.
Yeah, I mean, the first one was, I don't remember the results, but the cocktail was a little suspect. So the GH made sense, if that's your hypothesis. I believe, I've never spoken with Greg, but I believe reading the trial, the metformin, which was really given at a homeopathic useless dose, I think it was only given at 500, so apologies if it wasn't, but I think it was only given at 500, was meant to offset the glucose metabolism disturbances of GH. Do you remember why the DHEA was given?
No.
There was some reason for it that made sense on paper, but didn't make sense sort of physiologically. Now, the more important question is, if my take on that trial was it was a single active agent, which was growth hormone. Like, I don't think DHEA does anything. I don't think 500 metformin does anything. So the question is, and it was a very small trial, and I think it was open label.
And so I have a significant problem with the readout of that trial.
So that's what I wanted to ask you about. Remind me of the readout. The
readout was one of the clocks.
Ah, that's right. This was Steve Horvath was the other author on that paper.
Exactly. And actually this is this whole story sort of pushed us into a whole project that we've published on what we call entrant clock. Because there was indeed in the experiment, in the patients, they indeed observed some increase in the fraction of naive T cells, which tells you and me that something worked. The fraction of naive T cells increased with respect to the memory T cells. The naive T cells are the ones that are generated in the thymus. They're naive because they have never met their cognate antigen, and they sit there waiting for something to happen. So the whole idea of treating with human growth hormone was to induce thymogenesis and to restore the pool of these naive T cells. So I think to some degree it worked at low level. Then they used the clock on the whole blood. And my worry when I saw the paper, which is a worry that actually existed, predated this, it was also a worry when people were using telomere length, is the idea when you sample the blood, as an immunologist, I know this is a highly dynamic organ. Think about the blood as an organ. We enumerate at this point today with the best technology more than 500 different populations of cells in the blood. Suppose that these cells vary in response to any intervention and that these cells individually have a different epigenetic age. You would have the impression that you are rejuvenating, which was the claim of that Fahey paper, that they had rejuvenated people. But in effect, what you would do is simply change.
Yeah, it's like you're on a sine wave that goes like this,
yes,
and you take two sample points. They could be here, they could be here, they could be here. You don't. And by the way, as you probably know, Matt Kaberlin has famously purchased, I think, 4 or 5 of the commercially available aging clocks. He bought them in duplicate and did all of them, sampled them all simultaneously, right? So 2 of this, 2 of this, 2 of this, 2 of this, simultaneously. Take 10 samples. And not only do all the clocks disagree with each other, but even within the same clock, there was disagreement, significant disagreement. Yeah. I want to actually come back and talk about clocks in some detail. But given that that study was done years ago with an older clock, I think the clock part of it is not even remotely interesting. I think the more interesting question is, was there genuine thymic regeneration? If so, how do we reconcile a very pressing and vexing question within geroscience, which is the role of growth hormone? Right. So I've never taken growth hormone. I shouldn't say I've never prescribed it. I've prescribed it in very rare circumstances for injury healing. But I've never prescribed it for quote unquote longevity benefits. But a lot of people are out there doing so. And as such, I've had lots of patients who come to my practice who have been taking or are on growth hormone. And I will say this to a person, every single one of them has said, I feel so much better when I take growth hormone than when I do not. I mean, across the board, 100%. And I can't actually point to evidence that tells them it's bad to take. I can just sort of say it doesn't make sense to take if our goal is to reduce the risk of cancer and if our goal is to slow the aging process. So what is your take on that? Just your intuition, or is there any data you're aware of that would lead one to think that, well, maybe we could pulse a little bit of growth hormone here and there if we get some thymic regeneration? We don't have to be on it all the time. I mean, how would you think about that?
I do worry about it. I'm not a specialist on growth hormone itself. It induces diabetes. It induces glucose intolerance. So from that angle, I do worry about what it would do chronically, especially in someone young. For me, it's a bit like increasing your protein intake. If you are, there's that clear evidence that increasing your protein intake, especially as you age, becomes beneficial. And the people who have higher protein intake actually do better in terms of muscle mass and so on. So in someone who is 65 to 70, who is starting to feel, the effect of or manifests some form of sarcopenia, there might be a benefit for that person to actually increase muscular mass and all the benefits with this, especially if it's not done continuously.
But I mean, I would argue there's no doubt that there's benefits, but you're going to get far more efficacy from testosterone or anabolic steroids when it comes to mitigating sarcopenia. Growth hormone actually is not remarkable at inducing muscle mass. It's nowhere near as effective as testosterone. It's more effective at eliciting fat loss. But I wonder if there's something that goes beyond that. Because I think when people tell me they feel better on it, I think they're talking about like less aches and pains. Like just joints just feel better. I don't think anybody's saying they feel better because their thymus is more plump. But I wonder if like that to me would be a reason to potentially consider a schedule, you know, an intermittent schedule of something. If it's, again, going back to my macro thesis here, which is I've been harping on these four horsemen. Well, if we introduce a fifth horseman, what is the strategy? Because I can give you chapter and verse the strategy for how you will mitigate heart disease, cancer, all of these other conditions. What is our strategy for mitigating immune decline?
I would say the same as a strategy that would mitigate decline in every other organ. You know, and there's clear evidence that the effect of exercise on immunology is the same as in every single.
So I'm not familiar with it. So tell me a little bit about that. I don't know specifically how exercise impacts the immune system.
I don't know that, you know, I cannot speak to specific papers, but I mean, clearly there's evidence that people who exercise actually respond to infection better, respond to vaccination better. So that's all been documented.
I see.
I cannot speak to specific studies, but there is.
Do you have a sense of mechanistically why that's the case?
It is so complex, I would say. I would not be able to tell you. But that being said, I think the whole line of investigation to induce thymic rejuvenation, I think, is an important area, especially if we're thinking about increasing lifespan, you know, further for what we are doing now. That in the future, it will become one of these rate-limiting steps. It's a bit the same situation as the ovary, where, you know, these are the ovary and the thymus are, we call them the canary in the coal mine. I mean, there really are specific organs that show accelerated aging way earlier than other tissues. Now, the question is, you know, why is the thymic involuting so early? I think it's probably because evolutionary we were never meant to live this old, and so that really is one of the thinking that goes on. That's going to be in the long term one of the problems that we have to face. And this is something we're actively studying in the lab. We just completed a study where we are looking for novel biomarkers that are predictive of whether you will respond to a vaccination or not. And this is something done in collaboration with Mark Davis at Stanford using the 1000 Immunome Project, which is one of the largest studies studying aging in the immune system, only in humans. And so we've been able to studying people to identify some metabolites that are associated with poor response to vaccine. And so those are not only markers, but they could also become tools that we include as adjuvant or as a pre-treatment therapy. I'm sure you're familiar with the work of Joan Manick.
Of course. Yeah, I was going to ask you about Manick and Klickstein in a moment. But before we do, I want to go back to this point here, which is you know, biomarkers are so important. And when I think about cardiovascular disease, and even though it's the leading cause of death, why I tell my patients it's the one you need to be least afraid of if you're willing to be proactive in management. And it comes down to the fact that we just have such a clear understanding of how the disease works, and we have exceptional biomarkers. Right? So we can measure the things that are causing the disease. We can measure inflammation. We can measure ApoB. We can measure VLDL cholesterol, Lp(a). We can measure blood pressure. We can measure metabolic health, and we know how to address those things. And we know that when we address those things, we can measure whether what we're doing is working. Okay, so problem solved, basically. When it comes to the immune system, we're going to talk about Manick and Klickstein in a moment, but as we saw from their paper 10 years ago, they gave a rapamycin analog to people who were in their 60s, vaccinated them, and demonstrated that, oh boy, you got a much better immune response. Okay. They were able to demonstrate that using laboratory techniques. I'm sure they used flow cytometry or something like that to measure it. How close are we to being able to do that sort of thing commercially? By commercially, I mean over the counter, like
not close. I think in that study they actually measured antibody titers.
So even more complicated than flow cytometry.
Yes.
Okay.
And so in that case, they definitely showed an enhancing effect with a known, geroprotector. This was a
suspected geroprotector, at least in humans.
Exactly,
we hope.
A rapalog, and they showed not only increased titers but also increased protection. Eventually, the clinical trial failed for a whole series of other reasons, which were in part due to the way that the FDA imposed the trial to be generated. I think it just complicated the whole picture.
Yeah, by the way, for folks listening to us who are confused by that, Matt Caberlin and I had a specific discussion because it wasn't the 2014 trial. It was a later trial where it wasn't the RAD-001 trial. It was the other trial that failed. And I actually don't remember the reason, but Matt explained it. It was very clear that it was a tragedy of bureaucracy.
It is.
And it shouldn't be viewed as a black eye on that molecule.
Yeah, and I think Matt is more of a specialist in the whole rapamycin, so I will defer to what he said. But
we'll link in the show notes to where where Matt and I had that discussion.
Yeah, what I've heard from anyone that I've talked to, including Joan, is that this was in some way bungled, which is sad because sometimes things like this can put a field back for a number of years and discourage investors. We have a startup that originated at the Buck called Eovian, which has raised $50 million, again coming up with rapalogs, novel rapalogs that are going to be, I think, revisiting that whole picture. So we're quite excited. The field is far from being dead.
Will we ever be able to measure this in people the way we measure hemoglobin A1C or things like that? Or is it going to be one of those things where it's a bit of a leap of faith and you're going to have to look at the clinical trial where the outcome was there, and then you're just going to have to say, well, even though there was probably massive heterogeneity amongst the participants in the trial, we're going to dose this thing individually. I mean, it's a little bit like, you brought up vitamin D earlier. I mean, one of the problems with the vitamin D trials is that they're all garbage, right? Because they all just give people a given dose. They don't measure the response. They don't measure compliance. A vitamin D trial should be done based on target level, not target dose. And so we run the risk here of the same thing in a much more complicated system.
Agree. That being said, measuring pathogen-specific titers is done routinely in the clinic. I don't know if you did this, but I just had my measles titer measured because I was born in 1957, which is right the age at which before 1957 everyone was exposed to measles, so you are typically safe, but you should measure your titer to determine whether you need to be revaccinated. And I found out that, you can do this very easily. You get a titer.
But would the titers by themselves tell you, so what would you predict? So if I measured every titer right now, if I measured polio, shingles, did a pan titer on you, and then started you on rapamycin for 8 weeks and then stopped it and then remeasured your titers without vaccinating you, what would you expect to see?
I would not expect them to change.
Yeah, exactly.
No.
So how do we know we're improving your immune system if indeed we have?
I see what you're saying. So in terms of if we were to start you on rapamycin, what would happen? So
how could we measure the improvement in immune function?
By the way, the Manic trial showed that when first they did a 1 month treatment with the rapalog before vaccination. So this was they demonstrated not an effect on existing vaccinations, but only demonstrated on de novo vaccination.
That's right. Yeah.
And I think what would be the effect on existing titers against all of the other pathogen? I don't know. I mean, I don't think this has ever been done.
Yeah, interesting. You want to just say a little bit more about that trial? So that was, at least for me, that was a very pivotal moment in my journey in this space and in understanding this world. So that was December of 2014 that paper came out, and if I recall, roughly 300 plus participants divided into 4 groups. So placebo group, a group that got 1 milligram every day, a group that got 5 milligrams once a week, and a group that got 20 milligrams once a week. So two people were pulsed, one much higher than the other, and then one given daily, and then a placebo. I believe they were all over 65. I think the study was done in Australia. And as you said, they were put on their whatever treatment was for 4 weeks, immunized. I think it was another 4 weeks, and then a 6-week washout, and then the titers were checked. And the best response, I think, was in the 5 milligram pulse and the 20 milligram pulse. The 1 milligram daily still had a better response than the placebo, but not as strong as the 2 pulse doses. But the 5 and the 20 weekly were nearly identical, but the 20 had much more side effects. So I think that, and again, I don't remember perfectly, so correct me if I'm wrong.
You remember pretty well.
The takeaway was basically 5 milligram pulse was the sweet spot. You get all the benefit without the side effects.
Reason why, I think that's how I remember that trial as well. Although I'm always impressed by how you remember all of the details of these clinical trials. What was remarkable about that data was the fact that this is from a drug that
is supposed to be an immune suppressant.
An immunosuppressant. And, it's been a long road for the longevity field to try to get our colleagues who are actually using rapamycin as an immunosuppressant to have them believe that this actually has an effect on immunity. And not only not immunosuppressive, but actually promoting immunity. So
how do you reconcile that? Meaning not their disbelief, which is warranted, but how do you reconcile that one molecule can be, if given, so if you think about the doses we used to give rapamycin, it's not actually used that much, by the way, today in the transplant clinic. So FK506, sirolimus has largely, no, I'm blanking on what FK506's real name is. But anyway, whatever. It's largely displaced sirolimus, which is Rapa. But that said, when we used to give it out, we were giving 2 to 4 milligrams a day.
Yeah.
Now, let's just assume that it was indeed contributing to prevention of organ rejection. Do you think it was doing so because that's a high enough dose of constitutively giving a drug that it suppresses the immune system? Or do you think it was only suppressing the immune system because it was being given in combination with 2 other drugs, and it was only as part of that sea of other drugs that it has the immunosuppressive effects?
No, I think there's clear evidence it is immunosuppressive by itself. And I can tell you that for the period when I was on rapamycin, I would take either 4 or 6 milligrams a week. Mm-hmm. Every morning, so once a week. So the biggest difference between the two, the immunosuppressive and the sort of geroprotective effect is really the amount, the frequency and the amount. The reason why people adopted this sort of once weekly dose is to first not have any immunosuppression, and second, to mitigate the secondary effect, which are thought to be caused by inhibition of mTORC2, which is the second complex. Yeah, the glucose effect. And that seems to be working largely. What was, in my case, remarkable is that every time I took my dose, not 2, I only did 2 for a couple of weeks, but either 4 or 6, the next morning I would have a pimple on my nose. So I was immunosuppressed, clearly, every single time.
For a day?
Yeah, for a day or 2. And, I sort of made peace with it in the fact that if I had a really heavy workout, I would have exactly the same thing. Exercise is immunosuppressive. If you go all out, you can get a cold. You can, you're temporarily fragilized after a really heavy exercise. So I think the difference really between these two worlds, the immunosuppression, which clearly has been documented by clinical trials, it is immunosuppressive by itself, versus the beneficial effect on the immune system, to me is a question of dosage and frequency.
And yet, I cannot reconcile the unambiguous success of the interventions testing program, where those mice were eating rapamycin in every single bite of food they took. In fact, they were consuming it more continuously than even the most immune-compromised patient. And without exception, every single ITP study of rapamycin, whether they started in old mice or young mice, rapa alone, rapa with another drug, it just doesn't matter. It always worked. How do we reconcile that?
We do. Well, I don't have the answer, but I can sort of talk about it in a way. There's something that worries me about our reliance on the mouse as a model system for aging, for studying aging, and how relevant it is to us as species.
Even mice that are of the quality, because we would all admit that the ITP mice are the best, they're the Ferrari of mice.
Exactly. The ITP is the best because is the best way to address this question because they're using mice that are crossed.
Yep.
So it's not
inbred. Yep.
Not inbred. When you're using Black 6, you're essentially doing the experiment on N of 1. And the whole world, 80% of the work that's being done in mice is done on Black 6. We're all studying the same person. So obviously when you go and try to transfer this to a human population with all of its variation, so the ITP did the right thing. That being said, and this is not an attack on ITP. I think ITP is a great program and should be funded and we should continue to study this. I just worry about the over-reliance on ITP alone, and I think we should have another system that studies primate interventions with drugs. There are a number of primates, non-human primates, that are actually much closer to us. The reason I worry about mouse is something that actually Steve Ostad, that you've had on this podcast as well. Steve is a good friend, and he came up with something called the longevity quotient, which I think is something that people do not pay attention enough. So the longevity quotient is this idea that if you look across the animal kingdom, the larger you are, the longer you live. Okay, so you can take 1,000 species and you can weigh, on the x-axis you have their size, on the y-axis their life expectancy.
It largely rises to the right.
And you can see a monotonous curve. Now there are exceptions to this. One of them is naked mole rats. Naked mole rats, for example. Naked mole rats, are living way above. So
they live above. They punch above their weight. Dogs tend to punch below their weight.
Exactly. Although in dogs, it
varies by within the size thing.
This
it
is
seems
between
to hold.
species. Then when you look intra-species, it gets even more complicated, which is the larger dog lives shorter than the smaller dogs, the Great Dane versus the Chihuahua. And that is that's driven mostly by growth hormone, which is, again, another reason why we should look at, taking growth hormone as an anti-aging drug with some degree of circumspection, because in dogs, the more growth hormone you have, the larger you are and the shorter you live. We know also in humans, the larger you are, the taller you are, the shorter you live. So are these effective growth hormone? Yes. Are they only important during the growth phase? That's a possibility, but it's something that really gives me pause to go back to our discussion about growth hormone. Now, where was I? I sort of lost my train of thought.
We were still trying to make the argument for why ITP mice given rapamycin with every bite of food somehow live longer.
So again, going back to the respiratory quotient, the longevity quotient, mice are also an exception. They punch below their weight, so they live shorter than they should based on their size. And humans is the biggest exception. We live about 5 to 6 times longer than we should based on our size, which tells me that we are the naked mole rats of primates. We do incredibly well, which means that we already have optimized a lot of these pathways that are, promoting aging. I suspect the mice is exactly the opposite. I don't know that someone has really compared sort of intrinsic TOR activity in mice. Are they, for example, living in mice are, especially laboratory mice, are engineered to reproduce and grow as quickly as possible. They have large litter size. They do everything very quickly. Now, we know all of these activities are requiring a lot of anabolic, strength, which is driven by TOR. So the question is, are the mice sort of examples of animals that are maximizing TOR activity to do everything they do very quickly? And we are maybe at the other end of the spectrum, where we have low sort of basal TOR activity. So that's where I worry when people just transfer everything we know from TOR from mice into humans and saying it's going to show and work in humans. I don't know if you heard.
Such an interesting point.
Yeah. So I think this is frankly why I stopped taking rapamycin. I thought, I did not really see anything in terms of anything. Metabolically, physically, muscle strength, I could not, in contrast to GLP-1 agonists, where I saw all of my numbers get better, and functionally strength, all of this, I saw everything getting better on GLP agonists. With rapamycin, I never could tell whether I was taking it or not.
Yeah, although, look...
End of one.
Well, it's not just that. I would say where rapamycin acts, I don't know that we would see anything getting significantly better, right? Because if we think that the main places that rapamycin is going to act would be on autophagy, well, there's no way you're going to measure autophagy. You're not going to feel autophagy. You're not going to see it or measure it. Does it tamp down on certain subsets of senescent cells? That's certainly plausible. Again, I don't know how we're going to see or measure or necessarily even feel that. Does it reduce some of the tonic, low-grade, unhelpful inflammation? Probably. But again, if a person doesn't have much to begin with, it's going to be tough to measure. Conversely, GLP-1 agonists act directly on a thing that is so easy to measure, which is glucose metabolism and body weight for those who are losing weight as well. So it might not be a fair comparison. I guess the other thing I would add to this interesting observation is that, of course, the mice in the ITP are still in a relatively sterile environment. And it might be that even if they incur some immunosuppression, it's not going to be as maladaptive as it would be if they were wild animals, as we are.
They live in a sterile environment. They live grouped in a cage with no ability to move, to exercise. They eat a diet which makes the American diet look like the most healthy thing ever. I mean, have you ever seen the pellets that these mice are eating?
Do the ITP mice eat the crappy pellets as well?
I suspect they're eating...
Okay, I don't actually know what their diet is.
Yeah, I don't know what their diet is, but I mean, there are not that many... I can guarantee you they're not eating salad and fruits and vegetables. So in some way, they are an incredibly artificially bad, sort of environment. These mice actually doing everything that is conducive to a poor health. And so the fact that we see something that works in that system might have some value for the fraction of the population that has a very poor lifestyle. But I do worry about transferring this to someone like you and I who are exercising or trying to eat well or trying to sleep and all of this. So I take these observations with some degree of caution, and frankly, when people ask me should I go on rapamycin, I do worry. Now, this is a different, it's a very different story if a patient comes and sees you at 40 years old and tells you, I think I want to go on rapamycin. I would strongly argue that you should not do this, because even in the studies that have been conducted, they still saw an effect in mice that were the equivalent of 65 to 70 years old. Now, if you're 75 years old and you have the feeling you're chronically inflamed and you have the feeling that things are not doing well, there are a number of anecdotal cases where people have described really feeling a lot better and a lot stronger very quickly.
On rapamycin.
But I would predict it would be the same thing with a growth hormone or some of these interventions. So I really put those in different categories. And my argument to people is today we have one intervention that is very profoundly anti-aging, and it is physical activity, exercise in all of its forms. And once you have optimized this, I think let's talk about doing something else on top of that.
So earlier you brought up the sirtuin story and NAD. I'd love to spend a little bit of time there. I was at a talk recently, and as always, I get asked questions about stuff like that. And I got asked the question about NAD. And I said, look, this is one of those things where if I tell you the following facts, I'm going to tell you 3 facts. NAD is completely ubiquitous throughout the body, and It is absolutely essential for the most important chemical reactions that happen in the body. You cannot undergo redox reactions, metabolic reactions without NAD. That is point one. Point two is a class of proteins called sirtuins rely heavily on NAD as the substrate in the process of repairing DNA. That is fact two. Fact three is, as you age, NAD levels decline precipitously. Okay, those are three facts, and I don't believe, is there any dispute to any of those facts?
No controversy.
Okay. Armed with those three facts, how could it be that supplementing NAD does not lead to a longer, better life or some health benefit? That's a logical conclusion, right? Now,
well, not completely, because you are assuming, it depends also what is the reason why NAD levels decrease.
Yes.
And it depends also what supplements, or how you remedy this
yes,
decrease.
Yes.
And this is something I'd love to talk about, CD38 and some of the work that we've done.
So it could be that NAD levels go down because their consumption goes up, right? So there's as we age, there's more DNA damage, there's more consumption, the sirtuins need more of it, and it goes up. And then, of course, the question would become, is the current level of NAD that we have rate limiting to that reaction? If not, then all the extra NAD in the world should have no benefit because you're just adding more substrate to a reaction where it's not needed. Conversely, if NAD levels are going down because there's a production issue, and if you provided more of it, you could actually do more good, well, then it could be the exact opposite story. So let me pause there for a moment and have you fill in the edges of everything I just said so that we can go deeper into this discussion. So maybe explain a little bit what NAD is, explain, what it means in redox, and obviously let's talk about sirtuins and the role that NAD plays there.
Lots to unpack there. It could be a 2-hour podcast. But it's one area that we've worked on for the last 25 years. So we were responsible for cloning the human sirtuins, actually, after Lenny Guarente published his paper on SIR2 in yeast. We, with a graduate student.
Matt was the first to publish this, wasn't he?
Matt.
Caberlin.
Actually Matt and Brian, the whole, and David, that whole gang was the original gang, along with Lenny Guarente, were paved the way for a lot of what we know. Now, eventually, one thing that I would just start by saying is that it pains me in some way in a field that is so rich and has generated so much data that there's a whole cloud sort of lying on top of sirtuins and NAD, that, there's nothing there. I just tell people it is an incredibly studied system. We are still juggling the complexity. And I would argue that any field where the same degree of investigation will be conducted will have the same controversy. This is the nature of science. The beauty of science is that it's incredibly messy on the way up, but eventually things are getting clarified. And I think in the terms of the sirtuins, we're still right in the middle of it. So there's some complete garbage. I completely agree with you.
And by the way, I'm not completely dismissive. What I will say has made this field complicated is that the leading proponents of it have all opted for a commercial pathway.
Agree.
And therefore, they have opted not to study this in a rigorous way, but to study it in a commercial way. And unfortunately, I understand why you would do that. Like that's the nature of it. And this is not a molecule that you can put, you're not going to generate intellectual property in the same way that you would around a novel drug. And so it poses a limitation to how these things can be studied. But unfortunately, that coupled with the resveratrol fiasco, and I really, unless you think otherwise, Eric, we don't need to talk about resveratrol, but I remain completely convinced that resveratrol had zero benefit whatsoever.
Yeah.
I think it is an absolutely useless molecule. So I think that resveratrol debacle the overhype, complete debacle of that, coupled with the fact that all of the participants in the NAD landscape are doing it through their own commercial enterprise with their own proprietary blend, has resulted in this inability to drive forward in this field.
I agree. I think you've identified the problems, the hype and the commercialization. Commercialization can be helpful if the companies are actually willing to invest in clinical trials and so on. I always use the example of Timeline, urolithin A. We've worked with them. They do clinical trials, rigorous. They publish them in the best journals, and at the end, you know what you're measuring. And so that being said, for the sirtuins, so let's try to maybe sort of step back. And given the controversy, I would encourage your listener not to just discard it all. We're still in the middle of it. And I think there's something interesting will emerge out of it. So NAD is a critical intermediary metabolite. It has two big roles. First is it plays a key role in redox reactions. Again, we've talked about these reduction, oxidation.
Anytime electrons need to move around.
Exactly. So it exists in two forms, NAD, NADH, and it's critical to intermediary metabolism. There are more than 600 of these enzymes that use NAD in the whole metabolism. So it stands to reason that if you're losing NAD levels, it's going to be, you go below a certain critical level, these enzymes are going to suffer, your whole metabolism is going to go down. And we know, by the way, that decreasing metabolic efficiency at all levels is one of the hallmarks of aging. So in addition to these enzymes that utilize the NAD-NADH couple, there's a whole series of other enzymes that actually are cleaving NAD. And these would be the PARPs, polyadipuribose polymerase. So these are enzymes mostly involved in DNA repair. So it plays a critical role. Sirtuins, 7 sirtuins, all doing different things within the cell. And we can go back and dig into this a little bit in terms of what are the sirtuins doing. There's also two enzymes called CD38 and CD157. And so these are also NAD hydrolases, and we are studying them a lot. Now, so that's, I guess, the background of what these enzymes are doing. One thing that your listeners should know about NAD levels and why the decrease in NAD levels are relevant to aging is with respect to the sirtuins. Because sirtuins have a relatively narrow range of KD for NAD. So if the NAD levels change, as we know they do during aging, it will lead to a change in the activity of the sirtuins. And I think this is something that was proposed by Lenny Garante back in the days. It showed, for example, even during fasting, your NAD levels will increase, and this will activate sirtuin. So I think this is something that's really unique to the sirtuin. And we know this in a really acute way because there are sirtuins that are present in the cytoplasm versus in the mitochondria versus in the nucleus. And the NAD levels in each of these organs are very different. For example, much higher in mitochondria. It turns out that SIRT3 has a KD for NAD, which is much higher than SIRT1. And so variation, so that really is the indication that they are sensors of NAD levels, which goes back to the initial model that you mentioned. NAD levels change during aging, therefore we can expect the activity of the sirtuins to change. Now, what else have I not addressed in your initial batch of questions?
I think we're now ready to then move on to, if we believe with some conviction that restoring NAD levels in an aging individual is beneficial, we now have to deal with the same problem you deal with any small molecule, or large molecule for that matter. How do you get it in the body? So what are the ways in which you could get NAD into the body, directly or indirectly?
So that brings me to maybe another element of the biochemistry. Obviously, so one thing that has emerged is this idea that the question as to why do NAD level decrease? And there's been lots of theories. Activation of the PARPs, that seems to be happening in C. elegans, but in mammals, this is the work of Eduardo Cini, who was the first one to show that CD38 appears to be the major driver of the decrease of NAD during aging. And the way that he's demonstrated this, and we've actually repeated some of his results and published on this as well, if you study a mouse that's knocked out for CD38, you find that NAD levels actually do not decrease during aging. And that's pretty much across all organs. And I think that what this does is really brings the whole question is in terms of what should we be targeting. Now think about...
Did we talk about what CD38 is doing specifically?
Yeah, so CD38 is a membrane-anchored protein. Some of it is facing outward on the outside of the cell. Some of it is facing inward. For example, in T cells, it's mostly facing outward. In macrophage, it's mostly facing inward. And it is an NAD hydrolase. Now, what is it doing in the immune system? Why do we have it? Not entirely clear. One idea is that because it is present in T cells...
Is it on non-immune cells?
Endothelial cells
okay.
as well. One idea, at least for the immune system, is that it might come up and eat up all the NAD that's local in the extracellular fluid, although there's not much of it, and limit the abilities as part of the innate immune response and limit the ability of bacteria and other organisms to actually access these micronutrients for their own growth. That's one thinking. But I think it's a lot more complicated than this, and we're really in the middle of it. I have a good part of my lab actually studying the role of CD38 in the immune system and in endothelial cells and in the brain as well.
Now, you mentioned a moment ago that the CD38 knockouts do not see a decline in NAD with aging.
Zero, and they live longer.
I was just about to say, what is the phenotype of a CD38 knockout? What deficiency do they have?
Nothing that we can tell, and they live longer.
How much longer?
15%.
That's comparable to rapamycin.
Yeah, it's pretty significant. This has been published by Eduardo Cini.
Why do you think that is? In other words, do you think that that is true and unrelated to the increased pool of NAD?
Now, CD38, so that's a key question, and that's one that's not been answered. And I would say, if I had to go on a limb, I would say it's not linked to the NAD decrease. Because it turns out that...
Sorry, just to make sure everybody understands what you're saying. You're saying your belief is that the CD38 mouse, the knockout, does not live longer because he has more NAD. That's just another issue we're seeing, and that there's something else about that mouse.
Yes.
Okay.
Or it might be partially the NAD and partially the other mechanism I'm about to discuss. One thing that's remarkable is that as we age, us and mice, we see an increase in CD38 level across the organism, especially in the immune system. We've published a paper showing that the SASP from senescent cells is a very powerful inducer of CD38 expression in macrophages. So that's one mechanism by which we're linking, senescence and the SASP to increase CD38, leading to a depletion of NAD and other effects.
And yet we have no idea what it's doing other than hydrolyzing NAD.
It has a cognate receptor on other cells. It's part of the, they don't seem to be immune deficient. It's really one of these players that there are hundreds of papers.
I mean, do you think it plays a role in inflammation, a negative role in inflammation?
One idea about it is that it plays a suppressive role in the immune response. So because we see it being induced kind of late-ish in the immune response, and the idea it comes on to tampen down.
So it's actually the exact opposite. It's so pro-anti-inflammatory that it can be harmful in that way, as opposed to contributing to sterile inflammation, which is the more typical problem we see in aging.
Yeah, we did not discuss this, but the corollary, the other side of the immune system is that it has to be incredibly balanced between reacting appropriately towards exogenous pathogen, but not reacting against the cells. And as you know, as a physician, there are so many conditions that are a manifestation of an excess of immune response against the individual. All of the autoimmune diseases, which, by the way, increase during aging. So the thinking is that
Except for type 1 diabetes.
Yes, that's the one young.
I know that you used to study that. Why do you think that is?
It's really an interesting question. Although, there's something called LADA that I'm sure you've heard about that is emerging, that we might have been diagnosing some type 2 that were actually late type 1.
Yeah.
The thing that is really unique about type 1 is the fact that I remember a number of papers that highlight the fact that there might be something happening during development that exposes the immune system to the developing beta cells, and that might trigger more autoimmunity at that time. It could also be linked to the fact that there's been for many years a discussion of the role of viruses, infection, and molecular mimicry between some of these viruses and beta cells. So it could be that a subset of infections that happen during childhood actually puts you at risk of activating your immune system inappropriately.
Yep.
So that's the whole idea. But there's clearly an increased autoimmunity throughout life.
By the way, CD157, do we see the same effects? Do we have a CD157 knockout?
No, much less studied. There is some interesting effect also, but CD38 has garnered most of the attention. So if you think about, I'll go back about CD38 and in terms of what it's doing. It's taking NAD and cleaving it into ADP ribose, which is the sugar, and nucleotide, and nicotinamide. Nicotinamide is a precursor to NAD. And so this nicotinamide, which is generated by CD38, but by the sirtuins, by the PARPs, normally gets recycled in a two-step reaction all the way back to NAD. Now, what's really interesting is if you block this, it's called the salvage pathway for nicotinamide. If you block it, within a few hours, your NAD levels go down to 0. So the system...
Wow, heavily dependent on recycling.
Incredible, actually. And there's a specific inhibitor of this enzyme called NAMPT. You can add it to cells. We've done the experiment. Within 4 to 6 hours, NAD levels down to 0, the cell dies. So there's an incredible churning through that whole pathway. Now, which is a reflection of the activity of sirtuins, CD38, CD157, the PARPs, and so on. So you have a situation during aging where CD38 increases, you increase the degradation, and so you decrease the pool of NAD.
But you're increasing, in theory, the metabolites.
You're increasing the metabolites, nicotinamide. Now, one important thing is nicotinamide metabolism is either salvage back to NAD or methylation by an enzyme. And this is important for supplementation because it turns out CD38 not only cleaves NAD, but it also cleaves NMN, which is one of the two precursors, NMN and NR. So when you actually have increased CD38 activity and you take NMN, you churn through this pathway and you increase your nicotinamide and you increase its methylation.
So NMN is also cleaved by CD38?
Yes.
Into what?
Into
nicotinamide plus something that's not ADP ribose.
Yes.
Okay.
And so when you do this, you're increasing your level of nicotinamide to the point that it's shunting to methyl nicotinamide starts depleting your 1-carbon cycle. So what you see in a number of people actually on NMN is their homocysteine level going up, including me. I stopped taking it when I saw this. I thought I was taking about 1 gram of NMN for a while. And then I saw my homocysteine level going up, I think as a reflection of this pathway, and basically stopped it.
Now, why is it, Eric, that the increased pool of nicotinamide preferentially goes down a methylation pathway as opposed to the salvage pathway to give you more NAD?
Yeah, I don't think it goes preferentially. It just depends how much you're...
It's just the more you put in, even if it splits stoichiometrically or stochastically even, you're just you're gonna take away 1 carbon.
Yeah, and I do not know what the relative proportion is, but clearly the more you drive the system with NMN, the more you're going to yield these. And we can measure not only...
How much did your homocysteine go up, by the way?
Up to 15.
From
typical 7.
Wow, that's a big jump.
Yeah.
And then how long did it take to resolve once you stopped the NMN?
Well, I measure typically every 3 months. So after 3 to 6 months, it had gone back to normal, to 7 to 8.
What about NR, nicotinamide riboside? How is that treated by CD38?
It's not metabolized by itself. NR eventually in the cell has to make it back to NMN, which is on the salvage pathway that we talked about. So NR is less bulky, less big than NMN, so it is able to get into the cell, but eventually it makes it into nicotinamide NMN and then goes back into the same pathway. So eventually they all come back to the same.
So do you think that there's no difference between the same amount of NR and NMN?
No, there clearly are some differences, especially in all the really complex biochemistry that happens in terms of getting them into cells. The problem with NR and NMN is that if you think about what the approach is, you have a pool of NAD, which is a sink. Think about it as a sink full of water, and you keep, it's leaking. That's your CD38. That's the leak at the bottom of the sink, and you keep pouring more NMN, more NR inside of it. You're just gonna accelerate, the leak, or you're not going to solve the problem, basically. Maybe you'll reestablish the level at a normal, semi-normal, but the churning through is problematic. Now, why is the churning through problematic? Because some of the byproducts of CD38, for example, are cyclic ADP ribose. So there's two forms of ADP ribose, not cyclized and cyclized. The cyclized activates calcium signaling. And so there's a whole aspect of the biology of CD38 that's linked to calcium signaling. So I think I do worry about the supplementation with the NR and NMN. I do worry about it. I'm not discounting them. I think clinical trials are ongoing. There's dozens of clinical trials. So we will soon identify something in which it has a benefit. And again, if you think about the metabolism of these metabolites, it's incredibly complicated. There are effects on the microbiome. There are effects on different absorption by different cells. Just literature, hundreds and hundreds of papers, I think way beyond what your audience probably wants to hear. But and I would say, at this point, most of what you can buy as supplement have doses that are so low. This is where there's an important discordance also. When we do experiment, we've seen amazing things in laboratory animals in terms of supplementing with NR and NMN. This is where the excitement comes from. But typically, these animals are getting 10 times more than what you're buying as a supplement. And the reason is, I think, GRAS status is given to these companies to give a small amount.
Yeah, GRAS meaning generally regarded as safe. The
Yes.
FDA's criteria for giving something that is naturally occurring.
Yes.
Yeah, that doesn't require it to go down the IND pharmacologic pathway. Eric, if you were taking 1 gram a day of NMN and your homocysteine went from 7 to 15, I guess 2 questions would be, has that been reported elsewhere? Is that a known phenomenon? In the trials that are testing NMN, are they measuring homocysteine to see if that's...
I must have read it somewhere because
Okay.
I was
You were looking for it.
I was looking for it, yes.
And then the second question is, how would you then tolerate 10 grams of NMN? I mean, if 1 gram is doing that, now you would deplete all 1 carbon. What is that? Does that mean you wouldn't even be able to alter your epigenome in ways that might be favorable?
You run all kinds of risk. And, a number of people that I've seen the same thing and start taking trimethylglycine or, so to try to supplement this, I do worry about this.
Mm-hmm.
I think for me, and again, I want to reiterate the fact that I think the data in animal models of what some of the things that we've seen with some of these precursors is really interesting. And really, this is why there's so much interest.
Did you ever try using TMG to see if it would offset the...
No, I didn't.
Okay, that would be an interesting little self-experiment. Yeah. Okay, what about intravenous NAD?
So that is sort of one of my pet peeves. And again, I try in everything to remain open-minded to things that I don't know and don't understand. My prediction is that first, NAD is not an extracellular molecule. NAD does not exist, or almost does not exist at all in your plasma. It is an intracellular. And as I mentioned, high concentration in mitochondria, somewhat much lower in the cytoplasm and the nucleus. So the whole idea of injecting intravenous NADs, first, it's too big to be absorbed by cells. So what is the body doing with it? There is a famous paper by Josh Rabinowitz that showed that if you inject it actually intravenously, you actually get it mostly catabolized by the liver into nicotinamide. Now, nicotinamide is one of the fraction of niacin. You can buy this at the pharmacy for very cheap. You can go into an IV clinic and get a $700 injection of NAD. Very few studies, one or two. I've read both of them. They're interesting. So my opinion is that NAD intravenously is not something that should be done. The same thing for subcutaneously. I've seen another company that sells it subcutaneously. Really no evidence for doing this. Now, that being said, I've heard, and this is where I try to remain open-minded, because obviously we don't know everything. I have heard sort of anecdotal evidence of dramatic effect in some patients with Parkinson's. People really describing sort of not a miraculous, but a near miraculous effect right after the infusion, having increase in motor performance that you can really assess in someone who has severe Parkinson's. So again, not studied systematically. There's a lot.
Is there a reason you could explain that?
Yes.
Through dopamine or something else?
There's a whole literature on the effect of NAD precursors and so on Parkinson's, mostly in animal models. And I think there are lots of clinical trials going on in Parkinson's as well. So, but more using the standard NR and NMN. The thing that I've described is more a couple of friends who've told me, I've seen this. Not enough to make a product, but enough maybe to question, maybe there's something more to it than what we truly know. But, the proliferation of these intravenous clinics, frankly.
How complicated is it to produce a bag of intravenous NAD?
I don't think it's very complicated. I mean, I've never made it. I know making NMN at purity is really, it's not, took some effort to scale it up. And for some of the companies that have been doing this right now, there's like one major supplier out of China that pretty much everybody uses. But in terms of NAD, I don't think this is an industrial process. And I can tell you it's not $700.
Yeah, I'm sure it's not. So do you think, if a person was going to supplement with one orally, do you think there's a case for NR being superior to NMN?
I would say no. I would say take them both. If you're going to do something and you want a bit of an insurance, let's say, and I did this for a while. I'm not doing it right now. Take 250 milligrams of each, and you'll have a half a gram. You are in a relatively safe dose. Follow your homocysteine. If you are, 60 or above, you could make the case this could be part of a stack. Although this is the same thing that we see with so many of these supplements right now. Which one do you take? Which ones are beneficial? There's one, a little bit of a dark cloud linked to NAD supplementation, is the demonstration that the SASP is actually dependent on NAD levels. And so when you are actually increasing NAD levels, you might be increasing these pro-inflammatory markers.
So let me make sure I understand why. Because the SASPs, which for folks listening, these are the soluble products made by the senescent cells that effectively are doing all the bad things that we don't want to see senescent cells doing. So now they are dependent on CD38 to some extent. So as CD38 goes up, they go up. And are you saying as you give more NR and more NMN,
you might churn it up?
You might be churning up the SASPs.
Yeah. The other thing also is there's been some...
By the way, all of this... Let me finish. You finish your point, and then I want to make a broader point.
No, there's also some worry about the fact that supplementing with some of these precursors might also accelerate tumor growth. So this would not have an effect in you and I who don't have a cancer, but if it's someone out there who has an early form of a cancer, this could lead to an acceleration. This is something that's been shown in animal models that it giving some pause to some people in terms of recommending this to be taken over by everyone.
Now, the folks who make this have strenuously denied that there is any validity to those animal models that have suggested that, or and some of this has been done in vitro as well, correct? So I'm not very familiar with that literature. I remember seeing one study. It was very small. My take on it was, I guess if you had cancer, this might be a bad idea to take. But I didn't find it that convincing. So
I agree with you.
Yeah.
And it is a general consideration for our whole field of longevity research is, better is the enemy of good, is something that was sort of drilled into me as I went through medical school. We have a term in French, which is sort of like therapeutic overdoing it, just doing too much. There's such a thing as overdoing it for your patients. And in this case, the whole longevity field, is embracing a whole series of these interventions. I mean, there's not a week that doesn't go by that I don't see a new supplement being touted online and so on. And I read about all of them. The question is, which ones should you be taking? Which ones are actually risky? Which ones are not? And to me, this is part of the whole balance or the equilibrium that I'm trying to reach. If there's something that really has a beneficial effect, you want to be on it as soon as possible. If not, why take the chance?
Yeah, I think you said it. That's the point I was going to make at the outset, but you've said it so much better. Let's pivot a little bit to a couple other things I want to chat about quickly. Let's talk about interleukin 11. So big trial last fall that looked at blocking interleukin 11, which is a molecule that's made by immune cells, plays an important role in inflammation. And this was done in mice, and those mice lived longer. What do you make of the study?
Read the paper like you. I don't have sort of an inside knowledge about, of course, when the paper came out, it was like interleukin 11. I mean, as an immunologist, you talk about interleukin 1, 2, 4,
7,
6, 7, but 11, never heard about it. So I went to, and there's actually, it goes up to like 30. So I went and read the paper. It's an inflammatory marker. So again, it could be
on par with 1 and 6?
No, I would say probably not. So it came out a little bit in the out of the left field, but it sort of makes sense in the context of what we know about the inflammatory response linked to aging. So, and maybe this is where I can add one point is when we think about the chronic inflammation of aging, sort of inflammaging, it is both cause and effect. We talked about how the immune response helps you to protect yourself against the innate immune response against pathogen as a first line of defense. The innate immune response also has another important role, is that it recognizes damage, any kind of damage. If you cut yourself, if you have a wound inside of your organism,
coronary artery, a coronary artery. This will act any kind of damage, unfolded proteins. There's all kinds of things. So the innate immune response will be triggered and will activate itself. So as we age and as damage slowly accumulates, because aging is a slow, irreversible accumulation of damage, eventually your immune system responds to this by becoming chronically activated. And so the problem is that you might think, well, this is great because you're actually repairing all of this damage. The problem is the activation of the immune response by itself becomes problematic because these cells, macrophages, for example, are powerful tissue remodeler. And so the immune system in this case is sort of Dr. Jekyll and Mr. Hyde. It's helping, but it's facing an unsurmountable amount of damage. And eventually its activation leads to NAD depletion. That's one of the things that it does, but many other things, stem cell dysfunction and mitochondrial dysfunction. So the whole idea here is that
IL-11 might simply be, is one of the key markers of this chronically activated immune system. So this is not something I imagine you're going to give to a 20-year-old, but in someone who's getting really in the part where chronic immune activation is present, could really play an important role in the future. And what the paper showed was, again, in mice, but from what I understand, is already an existing molecule.
Yes.
And they're actually recently was contacted by a company that has another novel inhibitors of IL-11. You could imagine this to become part of the whole armamentarium that we have against aging.
And then how do you see playing that off something on the other side of the spectrum? Because we're really trying to deal with two sides of this system. We want to tamp down the part that's overactive, and we want to ramp up the part that's underactive. So we've got basically the only example we have over here is rapamycin. This one does this. And then we now have IL-11 inhibition, or not, they use knockout mice, but block this. That did good things. So is this one of those things where you need to do both together? Oh, and by the way, maybe you have growth hormone over here as well,
yeah,
right?
IL-1 there also.
Yes.
Anti-IL-1
That's right. So
as well, which has been shown to...
block IL-1, block IL-11, give growth hormone, give rapamycin. But here's the problem. You get into this reductionist state, which is like the whole NAD world of NR and NMN. Hey, it sounds great, but what if there's unintended consequences we can't see? Like, even as much as I love thinking about this and want to do all of these things, I start to think, man, what is the probability we're going to get this right?
Agree. And the immune system is an incredibly tenuous system, which is in really delicate balance. So the balance is too much immunity. You might say, well, this is good. Protection against cancer, protection against microbes.
Right, but then you get autoimmunity.
But then you get autoimmunity. Not enough immunity, well, you run the risk of being, killed by a pneumonia or some kind of infection.
But at least you don't have too much inflammation.
Yes.
Yeah.
So there's a very fine balance. And
well, this is why I wish we had a dashboard. What are the biomarkers we can use for these things? Because we don't have this problem with blood pressure. We don't have this problem with thyroid hormone. We don't have this problem with so many things that we treat because we can measure what we care about.
That's a good point. And the question is, what is, first, the immune system is so complex, there's not going to be one single marker. My colleague David Furman is this thing called iAge, which is a immune aging set of tests that you can actually conduct. That was the first attempt at trying to measure immune aging. They have a whole bunch of...
What do they actually measure? Is it all serum biomarkers?
Yes, serum biomarkers and mostly cytokines, CXCL9.
Validated how?
Been validated in clinical studies. So they've been...
IA, immunoaging?
Yeah. iAge.
Oh, iAge. Okay.
Yes. I don't know if it's so. This was developed and pioneered by David Furman and Mark Davis. So David Furman is with us at the BOC. Mark Davis is still at Stanford.
I guess this brings us to clocks.
Yes.
I don't even know if I have the energy to talk about this. Okay, where do you want to begin? There's so many of these things out there.
Yes.
Some of them are commercially available. Some of them are just tools of research at the moment. Some of them aim to tell you an actual age, an actual number that represents your biologic age as opposed to your chronologic age. Some of them don't aim to tell you that at all. They just want to tell you a rate of aging. Some of them look only at the epigenetic signature. In other words, they look directly at the methylation sequence. Others look at a host of markers, including some very simple serum biomarkers like glucose levels and vitamin D levels and things like that. So how do you make sense of all of those tools?
Right now, we don't. I think, so first statement is they are not ready for prime time in terms of patient management. There are research tools. And I think
Which is interesting because they're far outside of research labs at this point.
Yes, they are available commercially. I've done the same thing. I don't remember who told me. Someone actually, was it Matt?
Matt Caverlin.
measured his clocks. I do the same thing. Actually, I measure them every 3 months, and I use...
It's just a scatter plot.
It's a scatter plot in a way. I'm between 25 and 68, which is, of course, I like the clock that, show me to be young. But so that being said, so we know that, we're learning. So we know that, for example, you alluded to the fact that they can vary the same clock. There's circadian variation, for example, 5 years. So your age can vary by 5 years using some of the clocks, depending on when you measure, when you sample.
What time of day?
Yeah, what time of day. Which is, that's biology. That just tells you, the epigenome is something that's highly dynamic. And so that's something, as we learn, obviously, the companies will encourage you to measure it, to draw the blood always at the same time. Now, the whole field right now is pretty much focused, almost completely focused on DNA methylation. And Steve Horvath has done beautiful work. I mean, it's really pioneering work identifying and all this. And Morgan Levine and others have gone on. Dan Belsky, I think, with Denadine pace, which is another epigenetic clock that measures the pace of aging. By the way, I think this is probably my favorite because it really seems to be responding to interventions. If you change your diet or if you do something, you will see your pace of aging changing. So I think that one to me seems more promising. But it's a whole small... we don't know really how to use these tools clinically. That's the problem. They're nice gadgets to buy, and the companies are selling you supplements, and then they're selling you the tests with it. I don't know what to make of it. Personally, I think this is not ready for prime time. It's something that should be done in the future. It might become in the future.
Would you agree with my stern words on this? Because I've made a lot of enemies by saying that if, as a consumer, you encounter a company that is selling you a test, especially a test that is not validated in any clinically meaningful way, and then in the same breath selling you a supplement to fix the result of that test, you need to run.
I agree.
I mean, I don't have the patience for that kind of behavior.
Someone told me actually recently that one of these tests actually that you can measure, almost everyone who gets their result is low. And of course...
Low being good or bad in this test?
Or whatever. No, it's bad.
Bad, yeah.
It's insufficient. And the next step is the recommendation. You have to buy this supplement to solve the problem. So, yes, I think, again, it's the same thing with the sirtuins and the NAD. Let's not throw out the baby with the bathwater. There's a whole series of these players. I'm not disputing their honesty or their good intention. From what I've seen, I think it's too early.
And what do you think is the biggest problem? Is the biggest problem the biologic noise in the system, which means even if you had the absolute perfect tool to measure and you knew exactly what to measure, the movement of that thing is so great that the probability that you're capturing a meaningful value is irrelevant. In other words, imagine that there's a variable that moves like this, but on the small level, it's moving like this. And I'll give you an example. Imagine you were measuring heart rate, but you could only sample it milliseconds at a time, and what you were actually measuring was heart rate variability instead of heart rate. It would be useless. It's too noisy.
Agree.
So do you think that's the problem?
No, I don't think that's a problem. And I'll speak personal experience. I work with True Diagnostic. When you get, they use the Epic array and you get not one clock, you get dozens. So I get all of them. And they tend to be reproducible, every 3 months, unless I make some interventions. But in general, there is some consistency. And I'm not the only one who's seen this. So my advice, if you really are determined to use them, use all of them. They all are sort of different mirrors of your reality. The problem of the methylation clocks is that there's a very tenuous link between the change of methylation at any given site and the biology. So when, typically the clock will measure about 500, each clock would rely on about 500 different methylation sites, but they're not attached to a specific gene. So you don't really know what it means.
But how are they even doing that? They're not measuring with point arrays. How are they?
No, they do this with arrays.
They're doing this with an array?
Yeah, they have about 20 million methylation sites that they're assessed, but each clock uses a subset for 500. Now, the biggest problem with the clocks, from my perspective...
Sorry, just to be clear, you're saying they're actually measuring point of methylation?
Yes.
Okay.
Yes. They're quantifying the level of methylation at each of these sites. The problem with the clocks is also, where do you obtain them from? Typically blood, as I mentioned.
Yes.
It's a heterogeneous compartment. As you age, for example, you know that your fraction of naive T cells decreases.
Yep.
Down to close to zero if you're 80 years old. Your memory T cells increase. So we did a very simple experiment. We sorted all of these different T cell subsets: memory, naive, central memory, TEMRA, they're terminally differentiated, and measured their epigenetic age using several of the clocks. 20 to 25-year difference.
Between
the naive,
and
the central memory T cell.
In the right direction, the direction you would
Yes, the
predict.
naive are much longer.
Yeah.
So which is,
It's somewhat interesting.
That was really interesting for me, because it means also any conditions where you see a shift in the relative proportion of these cells. For example, you get an acute COVID infection. What happens? You have a massive expansion of your memory T cells. So it looks like you're going to, and then you sample, and given that these cells look much older than the other ones, you're going to look like you're aging. And so, and there's a whole literature that talks about accelerated aging in rheumatoid arthritis, in COVID, in HIV, all of these conditions that are all associated with chronic immune activation. So that's another confounding variable. So what we did to do this with a student in the lab, we made a new clock in which we eliminated all of these methylation sites that are linked to differentiation. Okay, so now this clock that we've done does not vary actually as a function of the types of cells that are in the blood. Okay, so as a T cell goes from being a naive T cell to being a memory T cell to being a TEMRA, the methylation patterns change. That's part of the epigenetic regulation. So we eliminated all of those sites and made a new clock called Intravenous clock, which actually is impervious to your level of immune activation. And what's interesting is that clock doesn't change anymore during COVID. It changed very little during HIV. It doesn't change during a whole series of conditions where people have talked about aging acceleration, including the story that we talked about earlier on growth hormone.
What does change it then?
What does change it? Cancer, senescence, which is really interesting.
What about short-term interventions that might be beneficial? So if you took an individual who is insulin resistant and you put them on a GLP-1 agonist, and 3 months later, they're 20 pounds lighter and their insulin resistance has resolved, how does that change on the clock?
Well, I would not be able to tell you specifically for individual clocks, but Dan Belsky's Denadin Pace clock is the one that repeatedly people have shown seems to be responding to interventions, which is, the two qualities that you want in a clock is one to be predictive, and the other one to be predictive of ultimate income, sort of life expectancy or the occurrence of disease, but also you want it to be modulatable, responsive, and many of them actually, but
and reproducible.
And reproducible, yes.
Yeah.
Reproducible, I think, is more a question of the laboratory that's doing it. So there are
but also potentially the biologic noise still.
Exactly.
So biologic noise and laboratory conditions speak to reproducibility. I agree with what you said. I mean, I've often made this case when people ask me about clocks is my gripe with the age clock. So again, the pace clock is different because it's just trying to give you a rate of aging, and I agree with you. I think there might be more there. But these clocks that spit out, hey, Eric, congratulations, you're 25. I say to someone who says, isn't that wonderful? I say, maybe, but do you actually believe that you're 68, you're 68 and your clock said you're 25? Should I expect you to live another 55 years?
Yes.
In other words, is it predictive? Is it a better predictor of future life than chronologic age? And the answer is, to my knowledge, no. There is no clock that has a better ability to predict lifespan than chronologic age does. And until that's the case, I worry that the biologic clocks are creating a bit of a distraction, at least this subset of clocks, and that we maybe ought to focus better on clocks where the readout state is more about, is this intervention good or bad?
Yeah.
Or is this a net positive intervention or a net negative intervention?
I agree. And as we mentioned earlier, the field initially focused on the epigenetic clocks because this is Steve's Horvath's pioneering work, so it got everybody to start thinking we can generate these tools. But the field is now moving into proteomics clock.
Mm-hmm. Is that so what makes up Dan's clock?
Dan is methylation.
It's also methylation.
Yes.
Why do you think it's doing a better job than maybe Horvath's clock at the moment?
I think typically it really depends on how the tool, what the variable, what the cohort, what the question was. I think I don't know. I mean, I think that Dan's is the only one that's doing it in this way. Why is it working better? They're just looking at it in a completely different way.
How much is AI facilitating this at this point? I mean,
machine learning is a key instrument. Essentially what these clocks are is a regression analysis onto, you start with a variable, which is your age, and you regress each methylation site onto the age. You do this on enough people of different ages, you find an average, and eventually what the clocks...
I wonder if that's the wrong way to do it. Wouldn't it be better to biobank these, to get biobanked data, and instead of mapping it onto age, map it onto number of years remaining in life? Because if you'll know that in a biobank.
They've done this.
Okay.
They've done this in terms of life expectancy. They've done this in terms of morbidity. So this is like the third and fourth generation of these clocks now are looking at regression not only on chronological. The initial one
was just chronologic age.
Yeah, and Steve used to go around saying my correlation coefficient is 99, and I was like, well.
That's because that's what you built it on.
Yeah, exactly. And
I
can look at a calendar. I don't need an epigenetic clock to tell me how old I am. But the next generation clocks actually had a bigger spread. When you look at, of course, you have an average.
That's right, because they started to build it on a different variable.
Exactly. Yep. So what really excites me right now is the whole idea that the field is moving on to the next stage, which is non-epigenetic clocks, because I'm still frustrated as a biologist trying to understand what are these clocks.
It should be everything. I mean, why would we do? It doesn't make any sense to me that we wouldn't look at the metabolome, the proteome, and the epigenome. I mean, we should look at every. I mean, there's no excuse today with the compute power not to do that, right?
We have clocks based on fundus. We have clocks based on skin. We have clocks based on facial recognition. So the clocks are going to be measured, using dozens of different biological variables. Any biome, a small company in the Bay Area is using a tongue picture, the old doctor looking at your tongue, so you can actually use machine learning to recognize patterns of discoloration and another, exciting really story was, I don't know if you're familiar with Tony Wiscoray's paper using proteomics. He has shown, for example, the proteome in plasma changes throughout life.
Yep.
Pretty dramatic matter, which is really completely mind-boggling for me to see that you can be so different as you age in terms of your whole blood proteome.
Why? I mean, wouldn't you if the epigenome is changing, then gene expression is changing. If gene expression is changing,
no, you would.
Yeah. It's okay.
But we haven't, so they've used this that it would change to such a degree. Tony has a beautiful slide which shows, all of the proteome in the blood and how the colors changed across age.
And do you think that most of those changes are post-translational?
No, most of them are probably expression level.
It's expression.
Yeah, it's expression.
Okay.
So people are building transcriptomics clock. And so Tony now has a study that is, I believe, in press or coming out soon, where they've gone back using this proteomics clock, and they've done this on a UK biobank, more than 40,000 different people. And this is the study we started this discussion on, identifying which organs are. So what Tony did was actually remarkable. He looked at each of these proteins that are in the blood and selected some that were predictive to be coming from unique organs. So imagine what you know about how you measure tropomyosin for heart attack.
Yep.
So they did this. They went and looked in every single organ and say, okay, what proteins are specific of this organ and which ones actually can be measured into the plasma. And using this, they were able to generate what they call an organ-specific clock. So what they, simply from a blood draw, they're able to really determine, do you have a frailty point? When I look at you, is there like suffering happening?
And this is Tony's work through the proteome. Yeah.
Tony was correct. It's a new startup called Vero, which is, full disclosure, I've joined the board of this company, but I only joined the board because I was really excited about what they're trying to do. And I think it really brings a whole new dimension to these predictive biomarker, which is more aligned to what you and I have seen as physicians. Just kind of simple-minded. If the protein is released into the blood, it shouldn't be there. It might be indicating some suffering. And I've discussed with some colleagues who have used these clocks and have identified some abnormal aging in a unique organ, only to go back and find that there was indeed one problem without going into, what the issues were. But you can...
Yeah, I mean, the reason I tend to be a slow adopter of these things is, even if that's the case, the question is how much noise is in the system. So I go and do that test on a patient, and it comes back and says, oh my God, there's something wrong with your liver, your kidney's a bit too old, you're this, you're that, you're this, you're that. So I have two fundamental questions. The first is, could I have figured that out another way? So if it's telling me your liver is angry or something's wrong with your liver, how do your transaminases look, right? If it's telling me something's wrong with your kidney, could I have picked that up on a urinary analysis looking at, creatinine clearance or, cystatin C or something else? In other words, is it giving me information that I can get elsewhere in a more reproducible, more validated fashion? The second thing is, let's say it tells me 7 things are not perfect. And and by the way, everything looks perfect. So I have my standard assays, everything looks awesome. This test says, oh my God, these 6 or 7 things are problematic. And I go poking around, and I find out one of them is indeed not working, but the other 6 were perfectly fine. So now we have this huge false positive situation.
It's the whole MRI.
Problem. Yeah, exactly. It's the same problem we have with cancer screening, which is, buyer needs to be aware of the Pandora's box you open. And at least with MRI, you're dealing with imaging. But this sounds like exciting, and yet it's a bit of a black box.
Agreed.
Where it's going to spit out, oh my God, there's something wrong with your left testicle. And oh my, what do I need to do? And
That being said, I think, the assays are generated in a way that there are multiple. It's not like one single protein, like a tropomyosin. We know that that's a clear indicator there's something cell death in terms of your heart. In this case, the clocks are generated in a way that there are multiple sentinels for each organ, many. Now, the story I was talking about, so early days, okay, we totally agree with this. It's a startup. I think they will... they will deploy it. And obviously it's going to take, again, a group of physicians who are able to look at these tests. And this is what research is. This is what startup, I can't blame them for trying, because I think it has a potential, for example, to highlight a frailty point, which is in aging research, to me, is really critical. You could have the best mind and the best heart in the world, if something else is going to fail that you are completely unaware of, you want to know as soon as you can. And so they my prediction is, I'll share a paper with you if you're interested in looking. It's quite exciting in terms of where this is leading. But I agree with you, early days.
Yeah, I will probably maintain a shockingly high degree of skepticism, and probably enjoy some experimentation with it. But again, my experience in the real world is that that's just not how it works, right? There aren't people walking around that are insanely, remarkably healthy where everything looks amazing, but they have some time bomb they don't know about, with the exception of a few things that I'm not sure it would pick up. For example, cancer is always that thing. And of course, there's an entire field of medicine that's going around with liquid biopsies that's exactly trying to solve that problem. It could be, you could reword the liquid biopsy industry through the lens you said, which is it's looking for that weakest link, which in this case is the earliest signs of cancer.
And it could be that a cancer will manifest itself also in local organ suffering, and again, leaching. It might actually point along with a liquid biopsy that tells you have some cancer cell. It might tell you, it might point you to one place where actually this is actually happening.
Yeah, again, it's interesting.
The case that I've made about MRI is the same, because I have a whole bunch of physician friends who tell me, I get a yearly MRI, and they tell me, why do you do this? I say, well, because I would rather know. And they say, well, you're going to find all kinds of things. I said, we did find something. I had a tumor behind my jaw, and it turns out a mass. It was not a tumor, but it took me 6 months of worrying about what it was, and decided not to biopsy anything. My sense of all of this is that these are novel ways to practice medicine. And I'm criticized heavily for being too much on the forefront of doing that, but probably not nearly as far as some. Right? I still, at the end of the day, I think about every time you do a test. One, you never do a test unless you're willing to act on an outcome, or you have a sense of how an outcome will change your behavior. We don't order tests for the sake of information. We order tests to make decisions. And therefore, you must, at a minimum, understand the full suite of outcomes that can come from the test and how many of them will pose huge trouble for you. I think, honestly, 20% of my patients opt not to do whole body MRI. Yes.
And I fully endorse that decision. And I try to talk patients out of it. I really try to highlight how many times we find thyroid nodules that we have to put needles into that ultimately end up being nothing. And all we do is subject them to that risk and the anxiety that comes along with it. So anyway, I'm eager to look at this, because I do think that the proteome offers a lot, but I'm always worried about going a little too far on the clinical implication of a test.
I agree. I'm with you in terms of being careful. I view this as another attempt. And for example, we talked about the data showing that the two organs that appear to be rate limiting in terms of aging, the immune system and the brain, that came out of that story. That's actually the title of the paper, essentially, that identifies the brain and the immune system. So they have a whole series of immune markers that are predictive of some degree of immune activation and so on.
Well, Eric, there's a lot of other things I wanted to chat about, but I think what we'll do is we'll have you come back out to Austin for another day of driving at COTA, and then we'll justify it by doing another podcast where we dive deeper into some of these topics. We really take advantage of the fact that we have the best race course in the country here in our backyard. So
absolutely.
I think you're going to have fun tomorrow, and
good.
You'll be like, let's come right back and do it again every month.
I'm looking forward to this.
Thank you, Eric.
Thank you.